Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Before we begin, a note from our sponsor. I'm Richard Jacobs, Executive Director of the nonprofit Finding Genius Foundation and host of the Finding Genius Podcast. In late 2016, I was rear-ended at 65 miles an hour by a truck on the highway, which sent me off-road into a ditch. The impact of the collision gave me a concussion and other injuries. At the hospital, a CT scan showed that I had thyroid nodules, which turned out to be cancer. It was then, when I had a biopsy of my neck, that I realized even if I was a millionaire, I wouldn't want a second or a third biopsy due to the pain and the invasiveness of it. And appointments at that time for thyroid experts were 3 to 6 months out. And I was worried about dying now, even if that was irrational. So because of this, I've decided to raise money to conduct a literature review on steroids, on the causes of anxiety and depression, a condition that affects well over 50 million people in the United States and hundreds of millions worldwide. Our goal is to create a codex, a guide that reveals all possible treatments for anxiety and depression for people that live with the condition, or for loved ones that have it, as my wife and my son do. To find out more about our fundraiser, visit findinggeniusfoundation.org and click on current initiatives. And now, to our guest.
Hello, this is Richard Jacobs with the Finding Genius Podcast, now part of the Finding Genius Foundation. I have Gaetan Burgio. He's a group leader, part of the Burgio Group, part of Australia National University. We're going to talk about AMR, antimicrobial resistance, and CRISPR. So, Gaetan, thanks for coming.
Nice to be here.
Tell me about your research. What are you working on?
So my research is divided in two different projects. So the first one is to develop CRISPR technology, so gene editing technology. And the second project is better understand why bacteria are resistant. So what we call antimicrobial resistance. Try to better understand why they are resistant and how they interact with the mammalian system, so the host, what we call.
So which bacteria are you focusing on? Are you looking at in what setting?
So I'm focusing on two bacteria that are quite important. These are causing a lot of trouble in the hospital. So the first one is quite known, is Staphylococcus aureus. It's a bacteria that has a huge effect in the hospital, in ICU and so on. And the second one, which is in my view more nasty, is a bacteria called Acinetobacter baumannii. So it's a bacteria that is resistant to pretty much everything, all antibiotics, and it's frequently seen in acid.
So Staphylococcus aureus, and what's the second one again?
Acinetobacter baumannii. Yeah.
Okay.
So the second one is less known. However, it is a more deadly bacteria compared to Staphylococcus.
Okay, where is Staph aureus? I've heard about it. I guess it's on skin, and there's a pathogenic form that can form. Is that the case? Where does it commonly appear? And for the other one, the Acinetobacter, where does that one definitely appear or commonly appear?
Acinetobacter baumannii is a nasty one because it stays everywhere. So mostly it's seen in surgical instruments if it's not properly sterilized, and is the one that is very hard to eradicate in ICU. So once it's there, the escape immunity, it is an absolute nightmare to eradicate, and it's resistant to pretty much all antibiotics. So it's way more nastier than Staphylococcus aureus in many ways.
So how do these bacteria, what happens when they become resistant? Do they take on extra genes or plasmids, or what have you observed?
Yeah, that's correct. So what happens is they acquire new plasmid, new resistant genes. They evolve quite rapidly, and they have a unique ability to persist, what we call persisting, so stay in the body, and escape immunity. So that's why it's very, very hard to eradicate those. And they acquire more and more resistance. So it's arm race between trying to eradicate those and avoid further resistance.
Okay, well, again, when they become resistant, how do they change? Is there, an underlying change in their genetic material? What's the mechanism by which they change?
So they can change all sorts of things. That can change their coat, they can change their metabolism, they can change their ability to take more plasmids, they can exchange plasmid with other species, they can do all sorts of things. And it's really, really difficult to eradicate those because they adapt very quickly and tend to change very often. So you think we have a treatment and then the bacteria evolve and change. We have another treatment, they evolve again and change it. So it happens all the time. So that's what they make them resistant to premature antibiotics.
Well, what's the mechanism? Has this been studied in vitro, and does it correlate with in vivo? When you introduce, let's say, bacteria in a dish, you introduce an antibiotic. Can you tell how the bacteria are adapting, the mechanisms of it?
Yeah. So the mechanism is basically the way they're adapting is they mutate. A good example, Acinetobacter baumannii is resistant to an antibiotic called colistin. So colistin is not, I don't know if it's employed in the States. It's not employed in Australia, but in certain countries they use colistin because this is the last resort antibiotics we have, carbapenem. So what happened to the bacteria? When we administer colistin in vivo, in the dish, it's the equivalent. The bacteria mutates its coat because the colistin directly interact with the bacterial coat, so what we call the membrane. And because it's mutating its coat, it becomes resistant to the antibiotics. And the bacteria has the unique ability to lose its coat, so it can live pretty much without a membrane, a large part of its membrane, which is quite unreal when we think about it. So there are multiple ways to escape the antibiotics. It can mutate, or it can clearly lose its coat. So it's one of the mechanisms. Another mechanism is to resist against the antibiotics. It pumps out the antibiotics when it comes into the bacteria. So it comes with a system called the efflux pump. So it's the antibiotics come into the bacteria, and the bacteria expel the antibiotic right away. So this is another mechanism, and there are plenty of mechanisms for the bacteria to escape the antibiotics. This is the way the bacteria survive and thrive within the body.
Oh, I've heard of efflux pumps. I've heard of all these things in the past. Okay, so are the bacteria that you study, are they typically in biofilms, or are they free living? And do they react differently if they're in biofilms? Do they acquire the resistance easier or harder, or what do you see?
So this is interesting because this is a bacteria that is not specifically virulent. So it's not the one that you've heard a lot. Yes, it persists in the biofilm quite a lot, but that is not only the case. Really, this bacteria has the unique ability to evade immunity. So that's why it persists a lot in the body, stay hidden for a while, and after when someone is immunocompromised, for example, someone in ICU, the bacteria becomes virulent. And really what makes really difficult to eradicate is the ability of the bacteria to acquire multiple antibiotics resistant, and secondly, to be able to escape immunity. Usually the immune system sees bacteria and eradicates it, not for that one, because it has multiple ways to escape. And this is what we study quite a lot in the lab.
I understand the immune system eradicates it, but again, if you have a biofilm, is it hardened? Is it more resistant to, are there trade-offs? Is it less likely to become resistant to biofilm, but yet it has more protection from the immune system, or are there no trade-offs? And this happens the same way with biofilms as with free-living bacteria?
Yes. So this bacteria, for example, has this ability to form biofilms. And once the bacteria are in community forming biofilms, it's much harder to eradicate because the bacteria are hidden, and so within the biofilm, and the antibiotics don't penetrate into the full biofilm, so they can't really get rid of this bacteria. So this is one aspect. The other aspect is this ability to escape immunity, as I was mentioning. So there are two different mechanisms of resistance.
Well, okay, so is biofilm formation just another way that bacteria become resistant? Could you count that in its cookbook of things to do to resist death?
Yeah, that's right. This is absolutely correct. And it depends on the bacteria. So some bacteria are more likely to form biofilms that are resistant to all possible detergents. Acinetobacter is a good example. in forms of biofilms, and biofilms are really hard to eradicate. And it fuels even more antimicrobial resistance. So these are typical bacteria that are likely to form a lot of biofilms. Some are less likely, and there are other mechanisms of resistance.
Before we continue, I've been personally funding the Finding Genius podcast for 4.5 years now, which has led to 2,700 plus interviews of clinicians, researchers, scientists, CEOs, and other amazing people who are working to advance science and improve our lives and our world. Even though this podcast gets 100,000 plus downloads a month, we need your help to reach hundreds of thousands more worldwide. Please visit findinggeniuspodcast.com and click on Support Us. We have 3 levels of membership from $10 to $49 a month, including perks such as the ability to see ahead in our interview calendar and ask questions of upcoming guests, transcripts of podcasts you're interested in, the ability to request specific topics or guests, and more. Visit findinggeniuspodcast.com and click Support Us today. Now back to the show.
Okay, so what mechanism of resistance do you focus on and study the most? Or are you looking at all of them and, what are you trying to accomplish and figure out?
Yeah, my focus is a lot on why the bacteria escapes immunity. Basically, why the bacteria thrives into a mammalian host. So what we call a mammalian host could be human or mouse, but most likely mouse, cause for obvious reasons. And basically, why these bacteria resist to antibiotics, escape immunity, thrives, is more virulent than ever. So I work on bacteria that are resistant to premature antibiotics, and I inoculate those in immune-deficient mice to see exactly what happened there. And I block multiple immune system pathways to see what happened there, if the bacteria can escape or not. And unfortunately, most of the time, the bacteria can escape.
Well, how? What is the mechanism by which they escape in some of these systems that you're setting up?
So we try to determine why. But basically, what happens is you inoculate the bacteria. The bacteria activates, let's say, one pathway, one immune pathway. You block that pathway, it activates, it escapes and activates another pathway. You block the other pathway. The problem is by blocking all possible immune pathway, the host doesn't have enough, I would say, beneficial inflammation to eradicate the bacteria. So the bacteria thrive and kill the host. So it's really what exactly in the bacteria is happening, I don't know yet. We try to determine what makes the bacteria escaping the immunity. That's the entire goal of my research.
Well, so we have no clue. I mean, is there different phage activity? When they become resistant, can you evaluate their virome? The phages that affect them.
So no. So yes, we can evaluate their virulence. They are not more virulent. It's just kind of strange. So they have this unique ability to escape the immune system. They are not virulent, but they use. I think so what the bacteria eat mislead the host, kind of make it think to the host, oh, okay, that's where I'm going, where in fact the bacteria goes to completely different ways. It's really bizarre. So we think that we have the right target to kill the bacteria, but in fact, even by targeting this specific molecule, the bacteria goes elsewhere because it misleads the host, and the host think that, okay, this is where it's critical for the bacteria, is the Achilles heel, and we can get rid of the bacteria, but in fact, it doesn't happen. And we don't really know what happened in the bacterial side. We try to determine this. Why is this so nasty? So why it does mislead the host?
Okay, well, any hints on the mechanism of how this happens and how it acquires these abilities to evade the immune system? What's the, any idea on how the bacteria is analyzing and, getting feedback and seeing how to adapt its approach?
Yeah, this is exactly what we're trying to do, to better understand why it does this and how. So we have quite an idea. So we have a bit of an idea. Basically, the bacteria mutates. So it mutates. It sees the immune system killing it. So what it does, it adapts and escapes by mutating itself. So that avoid the bacteria to get killed. It makes it less virulent, so the immune system doesn't detect it. That is another way of escape. So we try to determine exactly what's going on there.
What do you mean it becomes less virulent? I mean, how does it change itself specifically?
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
So this is trade-off between if it's virulent, the immune system detects it. If it's not virulent, the bacteria can persist, survive. So what happens is basically the bacteria tend to mutate some virulent component. For example, the efflux pump, as we mentioned earlier, to become less virulent. So this is one of the mechanisms, or it mutates its capsule. It does all sorts of things to avoid get killed.
Okay, so when it forms an efflux pump, it's busy with getting the poisons out of itself, and therefore it becomes less virulent because it's occupied, its energetic resources are tied up in that?
This is a possibility, but so the efflux pump is not only about antibiotics, there are other components as well. This is part of the secretion system, and the secretion system is the way for the bacteria to exchange metabolites to survive. So it can modulate its metabolite exchange, as I would say, to become more or less virulent. It adapts very quickly. It's truly incredible how those bacteria adapt.
Okay. Have you done this experimentation on single bacteria versus, multiple ones? Because I would guess there's bacteria to bacteria communication that says, all right, someone's in trouble. Let's help them out and give them this piece of code through a plasmid. They could develop an efflux pump, et cetera. Or are they developing this stuff just on their own somehow? Pump develops, or is resistance develops? Again, how does it happen? Like what's been observed?
So this is a very interesting question. So we haven't, unfortunately, done experiment with multiple strains of bacteria. The reason is practical, but this is something we came up quite a lot in the lab. At the moment, we do a single bacteria, but within the community. So within a single bacteria, it interacts with the host microbiota and can acquire more plasmids and so on. So it's not only the bacteria itself, it can interact with other bacteria too.
Okay, so you're looking at this in a microbiome setting in a host? Or, what kind of conditions are you looking at these bacteria to evaluate how they do what they do? Are they more successful in adapting when they're part of a microbiome of a holobiont? Or, under what conditions does this work better?
Yeah, so we trying to tease this out. At the moment, I don't have an answer right now for you. But looks like that the bacteria interact with the microbiota to acquire more genetic material.
Okay. Have you looked at the interactions of, let's say human cells with our microbiome cells, specifically, let's say Staph aureus or the other one you mentioned, and, to see what kind of metabolites are being traded, and maybe when there's resistance, the metabolites change that are normally being traded?
We haven't done that experiment yet. This is an excellent idea, an excellent suggestion. We haven't done yet. At the moment, We are in a single clone system because it's easier for us to tease out. When you introduce a bacterial community, that's much harder to determine what is doing what.
So how many bacteria will you look at one time? And again, do you have to control conditions so they don't form biofilms, or you just let them naturally do it? If they do it, are there any prompts to the system that cause them to, go into biofilm form or take on unusual forms?
So we don't necessarily look at biofilm. So we look at a specific part of the bacterial cycle. So when the bacteria invades, so the mammalian cells. So this is a really specific time. So upstream. So before biofilm formation, basically. So my colleagues in the field look at biofilm formation. I don't look particularly at this. I look more at how the bacteria is virulent, invades mammalian cells.
Okay, very good. Well, Gaetan, what hypotheses are you testing right now over the next year or so?
So there are two important experiments we are doing. One, antimicrobial resistance is basically really to get into what part of the bacteria is critical for immune escape. Which bit of the bacteria is the most critical for immune escape? Because if we know this, we will be able to target those antimicrobial resistance, so these AMR bacteria. So this is our main focus. And the second focus is to find treatment against those bacteria. So we are trying a couple of possible treatments as immune modulator that can eradicate those bacteria in what we call preclinical trial setting. So we have some candidate molecules. We are trying to see if they can basically kill the bacteria. That's the main focus right now.
So, since people can do single cell sequencing, would that be useful for bacteria? If you look at, a bunch of bacteria in a dish, and again, look at them in a single cell manner, and see which ones have changed and how genetically, and, maybe look at their transcriptomics, et cetera, and see in a population if they all react the same or they all act differently?
No, I think that would be very useful to look at single bacteria, which one are mutated or which one have not. Absolutely. This is a way to tease out what is happening to the bacteria.
Okay, well, very good. Gaetan, what's the best way for people to find out more about your work? Where can they go?
So on the website, the INU website, so I will very likely to update it very soon, and my Twitter feed. But it's all about CRISPR. A little bit about AMR bacteria, but mostly about CRISPR.
Okay, well, very good. Well, Gaetan, thank you for coming on the podcast. I appreciate it.
Okay, thanks for having me. It was a great discussion.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.